So we have the LacI minus mutant. Can somebody remind us what happens in a LacI minus mutant? What's wrong with it? What can't it do?
Induce transcription.
So what is LacI? Start simply. LacI is the gene that encodes LAC repressor. So this is a mutation to LAC repressor, which sometimes we also call LacR. But it's encoded by the gene LacI. So if it's got a minus mutation, what does LAC repressor normally do? What's its normal job? To repress. Go ahead. Binds the operator. Hello, operator. Right. LAC repressor binds the operator normally. So what does LacI minus prevent LAC repressor from doing? Keeps it from binding the operator. It's a mutation in its DNA binding site, so that here's some DNA. That's the operator, a specific DNA sequence, and you have some sort of mutant LacI minus protein that's missing its DNA binding domain. So maybe that's this surface down here. There's some sort of bump in it now because of this mutation. So it's missing its DNA binding domain. So it can never bind the operator, part of the promoter. So it can never repress. So what do we call this sort of a mutation? If transcription of the LAC operon is never repressed, it's always on, which is wasteful. Constitutive. Yeah, so this is a constitutive mutation. And we started talking about what happens if we can add back a wild-type version of the LAC repressor gene. Normally, this E. coli has a mutation in its LAC repressor gene that causes it to have constitutive expression. So what would we do if we added in the wild-type version? We discussed this last class. Just a quick reminder that by putting in a wild-type copy of LAC repressor, now that wild-type protein can do its normal job. It binds to LAC operator, it represses, and it can be dissociated from the operator. LAC repressor can be released from DNA how in this system? What causes LAC repressor to unbind DNA?
Lactose has to bind to the allosteric site.
Right. So lactose binds the allosteric site of LAC repressor and it falls off the DNA and then transcription can proceed. So that's how it restores wild-type function. We give it a wild-type copy of the protein. It responds normally to lactose. It can bind, unbind. You get a wild-type E. coli. And we talked at the very end of class about this bottom case, the OC mutation. So this is the second constitutive type. That's what that C stands for. That's also constitutive expression, always on. So if you don't have a mutation to the LAC repressor, what's the other component in this system that you could mutate that would cause no repression? You either affect the protein that binds the operator or you impact the operator itself, the DNA sequence. Right. So one of these, the LacI minus mutation, affects the gene that codes the repressor protein. The OC mutation affects the DNA sequence that LAC repressor binds. So they both have the same effect, that LAC repressor never binds the operator, in this case, because it doesn't recognize the DNA sequence at the operator. LAC repressor is looking for a specific DNA sequence. If that sequence is mutated, the OC mutation, then you get no LAC repressor binding. Transcription always happens. Constitutive expression. Now, what about the super repressor mutation? I should have worn my shirt. Maybe I should make a shirt. We'll sell them. We'll do a fundraiser. What sort of mutation happens to LacI that makes it super repressor? What does it mean that it's a super repressor, do you think? What's the function of a super repressor? What does that mean? it never unbinds the DNA. It binds the operator, and it never lets go. That's its superpower. It represses transcription of the LAC operon regardless of what's going on in the cell, that is, the presence or absence of lactose. Okay. So let's say we've got the E. coli genome, and here's the transcriptional unit, the gene that encodes the LacI with the super repressor mutation. So circular genome. Here's the LacI gene that encodes LAC repressor. This is the super repressor version. And up here on top of the genome is the LAC operon, Z, Y, and A. And here's the operator just upstream of the transcription start site. So we've got O as the operator, then we've got the LAC operon, and this is a mutant that has the super repressor version of LacI. So we just discussed that in this case there will never be any transcription because LAC repressor is going to bind at the operator and never let go. What happens if we add a plasmid, make a transgenic E. coli, we add a plasmid that has the wild type gene for LacI, I+. Okay. For a minute, talk to a neighbor, decide what you think is going to happen to this E. coli. Is it going to act like a wild type E. coli? Is LAC expression going to be normal, or is it going to be messed up still in some way? If so, how? One question and then we'll break up.
What does it bind to?
What does what bind to?
The LAC repressor.
So the LAC repressor always binds the operator. Super repressor binds the operator. Okay? So go ahead and discuss. Raise your hand if you've got a question. Yeah.
Sorry.
That's okay.
I have a question for the bottom one,
Yeah.
right? So there's a wild type there, so that means the transcription should be just as normal, right? Because even though there's a mutation for that one, the OC one, the
So
wild type.
look back at the notes for last class. We discussed why that's not the case. But basically it's because the plasmid that has the wild type version of O, that LAC-O plus
Right.
is not connected to the LAC operon.
Oh, okay. Yeah.
It's just the wild type copy of the promoter,
You might
and so LAC repressor can bind there,
interaction be
but it doesn't turn anything on.
because
I remember now.
Yep.
competitive stuff between professors.
It's like this. It goes like that.
The LacI and stuff, I just create a normal
All right. Seems like a good point to stop and collect your thoughts at this point. Do you have any questions before we get into what your responses are to this question? Concerns, uncertainties? I heard a lot of good discussions that were salient, pertinent to the crux of the problem here. It's hard to predict what's going to happen. Well, what did your group think was going to happen? What do you think the answer is? What's going to happen when you add wild-type LAC repressor? Okay, so we've got one vote for still repressed. Thoughts, comments?
Could there be, because if you have the wild type bind and there's lactose that comes in, is there a chance that because the wild type was there, the transcription could occur if the super repressor isn't nearby to replace the other repressor?
So this, So in this case, just like the top example, anytime we're dealing with the protein LAC repressor, that's transregulation. And that's, again, based on the definition of trans factors being factors that are freely dissociatable in the cell. If you add it in, it can act in different places in the cell. So this is transregulation, but it does not restore wild-type function. So the phenotype of the bacterium is more super repressed, even after you give a wild-type version of the LAC repressor gene. So you still get repressed transcription. Thoughts, comments, follow-up questions?
You said it would restore some function. Is there a level at which you could introduce the plasmid to restore it enough to be, I guess, discernible, or?
Sure. So there are ways that you could force the super repressor off of DNA. Mass action, for those of you that have taken biochemistry or certain forms of chemistry, if you put a lot of the wild-type version of the protein into the cell, and there's just a little bit of the super repressor, on average, when the super repressor happens to unbind, what's the protein that's going to then bind the operator? It's going to be the one that's in most abundance, which would be the wild-type version. So this is a very intricate process, and that's why I'm going to leave the rest of the discussion to a biochemistry enzyme kinetics class to talk about what are the relative efficiencies of binding and between the super repressor and the wild-type version and so forth. So the point was not to get into the mathematical details of the process, but just to point out that you can't always assume, this is the bottom line if you're going to write anything down, you can't always assume that giving an organism a wild-type copy of anything that's mutant, whatever it is, you can't assume that that's going to help. There are some situations in which gene therapy, for example, won't solve the problem, i.e. in the case of having a super repressor. A very sort of delicate situation. It's an excellent comment. So what's the dynamic between having a mutant version of LAC repressor and the wild-type version of LAC repressor in the same cell? So here's, let's say that's LacIs, the super repressor. And we've just added a gene that has the wild-type LacI, LAC repressor. What's the difference between these two proteins up there on the screen? Something about the allosteric site.
It allows the lactose.
So the wild-type version will bind lactose. So let's say lactose is a blue triangle. What's the mutation that makes LacI minus, or LacI super repressor a super repressor? What makes it always bind and never let go?
They can't be replaced by lactose.
It's got a mutation in its allosteric site so that it doesn't have an allosteric site anymore, or at least it doesn't bind its effector, lactose, anymore. Right? That binding of lactose is the thing that makes LAC repressor leave the DNA. So if you mutate the protein so it can't bind lactose in the first place, that makes it the super repressor version. So this is the problem then, which a lot of you were discussing. Here's the operator. We had LacI super repressor bound there initially, then we added in this plasmid that has the wild-type version of LAC repressor with the allosteric site. So what's going to happen to the bacterium? LAC repressor, unregulatable, the super repressor is bound to the operator. What happens when you add wild-type repressor? What did you say? LAC super repressor doesn't necessarily immediately unbind the DNA, right? That's the purpose, that's the function, that's what the superoppressor does. It binds tightly to the promoter and doesn't dissociate. So did our giving this bacterium the wild-type version of LAC repressor help? Not so much in this case. We really haven't changed anything. Now, I'm glossing over a few points related to enzyme kinetics. Don't throw anything. I know it's just past Easter, so I know you don't have any raw eggs. It must be hard-boiled eggs. What will happen in all actuality is that LacI superoppressor doesn't have perfect affinity for DNA. That is, it is not irreversibly bound to the DNA. It just likes to bind there. So every once in a while, yes, by chance, that superoppressor molecule will dissociate. So maybe the bacterium is in an environment where it warms up during the day, and there's more energy in the system, and the proteins are binding and unbinding the DNA a little bit more frequently than they normally do. Then the wild-type version of the protein has the chance to bind. And then you get somewhat regulatable transcription based on the presence or absence of lactose. So bottom line, though, is I would expect all of you to say, and as you were saying, in general, LacI superoppressor just binds and prevents any sort of regular activity in terms of transcription of the lactoferrin. Switching gears a little bit, you've watched the video for eukaryotic gene expression regulation for today. We just need to fill out this table really quickly. In which sort of organism is, in general, again, this is overly simplified as it says up here, which sort of organism is it where transcription is usually on unless it's turned off? This is prokaryotes. So then you better know what's on the bottom line. Eukaryotes. What I haven't discussed and what I didn't mention in the video that you watched was why this is the case. So what's the difference between prokaryotes and eukaryotes? We've already talked about this in class. It's been a month or two. What's the difference between prokaryotes and eukaryotes that causes this pattern? It has something to do with their genomes. So heard shape, circular, linear, not quite yet, close. It does have to do with chromosome structure.
Is it because things like prokaryotes are trying to be as far as possible, so it just always has to be on?
What is it about, it's maybe related, but what is it about prokaryote chromosome structure that's different than eukaryotes?
Prokaryote structures are usually more open versus eukaryotic will have condensed structures.
So eukaryotes, remember, have chromatin structure. What's chromatin made up of? DNA plus proteins, and the proteins are histones, right? Our DNA is normally packaged into condensed structures. Sometimes it's open, sometimes it's condensed, but that means that eukaryotes need to be able to open up the DNA structure to have transcription. So our default, and again, this is overly simplified, this is not true for every gene in every organism. Our default is our DNA compaction keeps transcription from happening unless we have activated proteins that come in and open up the DNA and allow transcription to occur. Prokaryotes don't do that. Their DNA is just naked, sitting there in the cell. Transcription's always happening, and that's why they need proteins that are repressors to turn down transcription in situations where the bacteria don't need transcription to be occurring. A whole different pathway today. It's still a pathway having to do with the utilization of sugars, but in this type of situation, we've got eukaryotes in the galactose pathway. I don't want to go into the details of the protein mechanisms because that was in the video, but I did ask you to finish filling in the repression and activation pathway drawing. So work on this for a second. You just need to draw in the blunt arrows or the arrow heads between these different components. So I'll give you 2 or 3 minutes to work on this. Feel free to collaborate if you want, and then we'll come back together and I'll ask you to share what the pathway is that controls, starting at galactose here and winding up at the expression of gal1. So go ahead and work on that for a minute or two, and we'll come back together. Again, raise your hand if you've got any questions. Review your notes, refresh your memories. Questions back here? Okay. What was the part of this pathway that was in the video to start with? Question? Yeah.
Aren't the 2 4s and the big one on the bottom 80?
Yeah, it should be the other way around.
Yeah.
Gal 4 on the.
Gal 4 UAS. Yeah. Gal 80. Glad people are paying attention. So what does Gal 4 do to Gal 1? Gal 4 activates Gal 1 expression. Gal 80 does what to Gal 4? Right, so that's what's happening here. Gal 80 is covering up the activation domain, AD. Like most regulatory proteins, there are 2 domains, a DNA binding domain and an activation domain. Okay, so what does Gal 3 do to Gal 80? I haven't diagrammed the protein, Gal 3. So what do we want to have happen when galactose is present? We want galactose to induce the transcription of Gal 1. So galactose somehow should activate these genes that are necessary for a cell to use galactose as a carbon source. So that's the logic we're trying to get to. We're trying to figure out how galactose activates Gal 1. So together, galactose and Gal 3 have to do what to Gal 80 to turn on?
Wouldn't they Gal 80?
It has to repress. Because there you get that double negative that makes a positive interaction. So Gal 3, when in the presence of galactose, galactose binds to Gal 3. So let's again assume that the sugar is some sort of a triangle shape. So we've got galactose, we've got Gal 3. Together they pull Gal 80 off of Gal 4. So it represses Gal 80's normal function, which is to repress Gal3, or Gal4. Right. So that's why that's a repression event. Gal3 and galactose together prevent Gal80 from doing its normal job, which is to repress Gal4. And in turn, that allows that activation domain to bring transcription factors to the transcription start site to the promoter of Gal1, you get transcription. So that's the rest of the Gal pathway. Just a quick review. I wanted to make sure we got to cover that. Now, how does any scientist study whether or not transcription is happening in a cell? I want to get into a little bit of experimental detail about what we do in the laboratory to measure whether or not transcription is happening. Because it's easy to say, yeah, transcription turns on, transcription turns off, but what does that mean when we actually try to study that? And so I'm going to use as a case study this protein that we've seen before. Does anybody remember what protein this is? This is the TATA-binding protein, a eukaryotic protein, a transcription factor that binds to which DNA sequence? TATA. Right, that's why it's the TATA-binding protein. It binds and recognizes the DNA sequence TATA, and it's one of those transcription factors that brings RNA polymerase to the transcription start site on a DNA molecule. So this is TBP, crystal structure bit bound to DNA. I've shown that before. And we've also talked about the fact that TATA-binding protein has this sort of saddle structure where it binds to DNA, and when it binds DNA, it actually physically bends the DNA. And there are amino acids in TBP's stirrups. Those amino acids actually physically contact the nucleotides that TBP is reading. That's how it senses what the nucleotide sequence it's binding to is. It's by contacts between those amino acids in the stirrup domains, those loops, and the DNA sequence. So channel what we just talked about in terms of prokaryotic gene regulation and the LacO mutations, like the OC mutation. What would happen to transcription to us, or to yeast in this case? This is the experiment that I'll show you in a minute that was done in yeast. Another eukaryote. What happens if you mutate the TATA sequence? What happens to transcription?
There's a chance it won't happen.
There's a chance it won't happen. What's TBP's role, interaction with transcription? Is it positive or negative? Positive. It's an arrowhead. The binding of TBP turns on transcription. So if you mutate the TATA sequence into, say, TGTA, all of a sudden you've made a sequence that TBP won't bind because it doesn't recognize it, and then transcription, we would predict, would turn off of whatever gene this sequence is upstream of. So what we're going to look at is situations where we have, instead of TATA, we have TGTA in the promoter. And there's going to be some gene, and we're going to try to figure out whether or not it's being transcribed. This is the purpose of a reporter gene. And all this is a pretty simple concept. This is geneticists making recombinant DNA, that is, combine a specific promoter, like a mutant promoter that we have here, with a specific gene that's easy to measure transcription of. That's all it means when we talk about a reporter gene. It's just a convenient, it's a protein that does something easy to measure so that we know whether or not transcription is occurring or not. And I hesitated on whether or not to include this because I want to get people, we'll see in a couple slides. I don't want to confuse the situation, but we'll see what happens. For TATA-binding protein in yeast, it was known that if you make this mutation TATA to TGTA, right there in red, that reduces transcription. So that's a fact. You predicted that correctly. What's interesting is that then scientists working with that TGTA sequence realized that they could find mutant versions of TBP that prefer to bind to TGTA. And that's what's listed down here. These mutant TATA-binding proteins have compensatory mutations. So what's happening, and we've looked at this picture before as well, this crystal structure of TBP up here bound to DNA, which is right there. There are two amino acids in the stirrups down here at the bottom. There are amino acids 194 and 205. Those are the ones that really make contact with the TATA sequence. Those are the ones that specify what sequence TBP is going to bind to. And if you change those amino acids to different amino acids, then you can change the DNA sequence that TBP recognizes. So that's what this second bullet point means. So you make two mutations to those two amino acids. You change here an isoleucine to a phenylalanine at position 194, and you change a leucine to a valine at position 205, and all of a sudden, you've made a TBP that doesn't like TATA, it likes TIGTA. I'll just stick to calling it TGTA. Separately from that, so that's one thing. We can have compensatory mutations that happen in the protein that help it recognize new DNA sequences. Then this other weird thing happened, the third bullet point, which was somehow the scientists discovered that if you take off the N-terminal portion of the TBP protein, the whole start of the protein, if you get rid of it, then TBP, TATA-binding protein, loses its specificity for TATA. So that means that not only are, briefly, it just means that not only are these two amino acids, 194 and 205, important for recognizing a DNA sequence, but so is the N-terminal domain. So if you delete the N-terminus, that's what that triangle N, delta N, or deleted N-terminus, then TBP will bind OK to TGTA. So two different things. Point mutations in the stirrup and the presence or absence of the N-terminus dictates whether or not this protein will bind and start transcription. So when I was an undergraduate, this was the task that was given to me in my first research experience at University of Oregon. My faculty member that I was working with said, I'd like you being me, to make a mutant version of TATA-binding protein that combines that absence of the N-terminus with those two point mutations. So what do you predict would happen? So we've got 4 different versions of TBP. We've got the wild type version, we've got the N-terminal deleted version, we've got the double point mutation, isoleucine to phenylalanine and valine to leucine. And then we've got the 2 mutations combined. The N-terminus is gone, plus there's 2 point mutations. What I just said was that with the TATA sequence, wild type binds fine. And if you have any of these mutations, those TATA-binding proteins won't bind TATA. Wild type TATA-binding protein won't bind TGTA. So what happens to these other 3? Delta N, this is review. So you get rid of the N-terminus, you get transcription. If you do the 2 point mutations by themselves in TATA-binding protein, you get transcription. So that's the question mark, is what happens when you take those 2 mutations, would we get 2 plus? Do we get super transcription? Pardon? Yeah, that's the question. Okay, so this is where reporter genes come in. I need some way to measure whether or not transcription is happening. So the way this works is you have the reporter gene, here's the transcription start site on a plasmid that we can put into yeast. So up here in the promoter, one of two things happens. We either have a TATA or we have what? TGTA. So one variable is which promoter do we have? Do we have the wild type, TATA, or do we have the mutant version, TGTA? Downstream of that, we have, this is the part that I'm going to be extra careful to make sure it's not confusing, the gene that encodes the enzyme beta-galactosidase. Beta-galactosidase is an enzyme. All that enzyme does is cleave a particular type of chemical bond. This one in blue. So it breaks that bond. And what happens is normally beta-galactosidase is used in what situation? When there's galactose, beta-galactosidase breaks it into two simple sugars. That's like the GAL pathway we watched the movie on today. GAL utilization. In this case, we use beta-galactosidase not because it has anything to do with what you watched for class today, but because it does this chemical reaction with a really simple substrate here, ONPG, which is a derivative of galactose. The value of using this is this molecule, ONPG, is clear, but when beta-galactosidase is present, what happens? It cuts this molecule into ONP, orthonitrophenol for you organic chemists, right? There's the phenol, there's orthophenol, orthonitro. That is yellow in solution. And galactose is a sugar, it's clear when it goes into solution. So why is this useful? Now I can see transcription visually because if TATA-binding protein is binding one or both of those, transcription of beta-galactosidase occurs, translation of the messenger RNA into protein occurs, I've got beta-galactosidase, and then I just throw in some ONPG, literally, throw it into the tube. And if beta-galactosidase is there, you get this reaction from left to right down there on the bottom. You go from clear to a little bit yellow to a lot yellow, and the amount of yellow tells me how much transcription is occurring. The amount of transcript that's produced correlates to the amount of protein, beta-galactosidase that's produced tells me, is transcription working with the mutant TGTA promoter or the wild type TATA. And here are the data. So again, the point of this is just to understand how the use of these reporter genes works, why they're useful. So in this case, all I want to know from this slide is, was our hypothesis, what was the answer? What happens when you combine those two mutations? What happens to transcription? So this was that new mutant. N-terminal deletion plus the 2-point mutations. It's got the highest level of transcription with the amount of yellow color being plotted on the y-axis. So more yellow is up here. It's more transcription. So yeah, combining those two mutations made TATA-binding protein even better able to bind to TGTA, which is pretty interesting. So I was basically engineering proteins to bind new DNA sequences. Kind of fun. And then I went off and did totally other things after I got my biochemistry degree. But so this is reporter genes and why they're useful. Something simple like you can monitor the amount of yellow in a vial, that tells you something about the amount of transcription. And TATA-binding protein plays a key role in this process. end up, I was really inspired by the presentation we had at the start of last class. This protein, cry1, we were talking about, which I had never heard of before, but I was inspired to go read the manuscript on the presentation we heard. And remember, what's happening here is we've got people that have weird sleep patterns, or non-normal sleep patterns, I guess. And that has something to do with mutations in the protein cry1. It doesn't have anything to do with crying. It stands for cryptochrome, but that's not important. And people that have, what's that triangle mean again? There's a deletion. So here we're talking about another protein that's got a deletion in it. Somehow affects our ability to regulate our circadian rhythms. So I want to send you this Google Classroom activity. So here it comes. And this is what we're going to end up doing today. So hopefully you start seeing that in your feeds pretty soon. So what I'd like you to do, and we're going to continue this next class, is to think about, so when you read the instructions, it says there's a gene A and a gene B that make protein A and protein B. And what does it say about abundance of these proteins? What are we supposed to assume at time 0? There's, sorry, about?
Because A is abundant and molecule B is not present.
So we've got a bunch of A, and we don't have any B. So that's the starting situation. So what I'd like you to do is follow the instructions there and just pick one of those 4 simple genetic pathways on the right. 1, 2, 3, or 4. Just pick one. And then maybe work with a partner. You can do it by yourself and then compare notes for the next 5 minutes. Just draw out over time what you predict is going to happen given the relationship between gene A and gene B. What's going to happen there, the amount of those 2 proteins over time. So make that prediction, draw it out, send it back to me through Google Classroom when you're done, in 5 minutes or less. Just, accuracy is not important here, I just want to know what your prediction is. And then we're going to talk about your responses. So go ahead and work on that. Come back together in about 3 minutes for a quick wrap up. So the goal is to get this turned in by the end of class, and we'll start talking about your responses starting next class. Plot over time, yeah, what's going to happen to the amount of A and what's going to happen to the amount of B. That's fine. Sure, yeah.
Okay, B, I'm gonna put the color.
Questions? So please turn those in when you can, sometime before next class. I'm going to flip ahead last slide. I'm going to go over all of these slides, the rest of today's presentation on Friday. But what we're really aiming to get to on Friday is to talk about something that has to do with many of you, maybe, in transcription regulation, how we wind up going from milk-sucking infants to adults that have this happen to us, lactose intolerance. So there are important transcriptional changes that are involved in whether or not we remain lactose tolerant like we were as babies, or whether or not we get bloating, cramping, diarrhea, etc. So come back for that on Friday. See you next time.